This video is part of a series. It's heavy in scientific terminology and may expect familiarity with previous videos. If you're confused, please watch the rest of the playlist up until this point, or check out my genetics terminology document, both of which are linked in the description. Thanks! The term dilution in genetics refers to when a gene reduces the density of pigment across individual hairs or skin cells, causing them to appear lighter. You ever see one of those old comics that's colored with a bunch of dots? The dots are red, the paper is white, you zoom out, and it looks pink. Kinda like that. In dogs, a given dilution gene will either affect only eumelanin hairs, aka black base markings, or pheomelanin hairs, aka red markings. So a dog might have diluted black base markings but dark red markings, or they might have diluted red markings but undiluted black base markings. If both are diluted, it's because multiple dilutions are working on them at the same time. On wolf grey dogs, who have banded agouti hairs of both eumelanin and pheomelanin, as well as on sable hairs which are also banded, only the corresponding bands of hair on each will be affected. So a wolf grey dog without any dilutions will have an agouti hair that looks like this, with just eumelanin dilution, like this, and with just pheomelanin dilutions, like this. In both of these latter cases, it'll cause the hair to look lighter overall, but less so than if it was fully lightened. When someone talking about dogs says dilution, they're almost certainly referring to the mutation in the MLPH region that only affects black base. Or, rather, mutations, plural. There's D1, D2, and D3 was recently discovered by a study done in 2020. The article that found it says that there are probably even more distinct dilution alleles that we've yet to find. We don't know about them because they're all basically identical, so the only way to tell the difference is to do a bunch of expensive, complicated genotyping on a hunt for them. That means that once again, this is a situation where a bunch of alleles do the same thing and inherit the same way, so we pretend they're all the same allele. For simplicity, we lump all of these D numbers together as little d. It's recessive to the non-dilution allele, capital D. If the dog's eumelanin hairs showed black, then homozygous little d will turn it blue. If the dog's eumelanin hairs showed liver, then homozygous little d will turn it Isabella, also known as lilac or lavender. This is another one of my favorite dog phenotypes. It's so beautiful. This dilution affects all eumelanin across the whole dog. Any dominant or recessive black, brindle, black markings, masks, stray tipping on a sable, and even nose, paw pads, eye rims, and whiskers will be affected. Keep in mind that since a red dog's leathers still show their underlying base, a clear sable or recessive red dog's nose will still show whether they're dilute or not, even though their fur won't. Interestingly, liver noses can get as light as Isabella noses despite the lack of dilution, so this trick for figuring out a red dog's color won't always work. There's a slim chance of misdiagnosing a liver as an Isabella, though Isabella sometimes has a slight purpleness to it that liver doesn't. That all being said, this dilution does technically affect red too, but not very much. Red already has so much variation in shade due to its own modifiers that you would probably never be able to see the tiny little push that this dilution has on it. Just know that because there is some sway, if a dog's dilute, their red won't get too dark, but it can still get pretty dang dark. Just like liver, dilution causes a dog's eyes to become amber. I can't find concrete info on if the dog is both liver and diluted, aka an Isabella, but from pictures they seem to just have like a paler amber. There's a genetic disease in dogs called color dilution alopecia, or CDA for short. CDA causes hair loss and the inability to grow more hair, itchiness, scaly skin, and makes a dog susceptible to a bacterial infection. It can look like mange, but mange is totally unrelated to CDA and is caused by mites, not genes. Thankfully, CDA isn't deadly, it just sucks. There's a myth that the D locus, or being dilute, pleiotropically causes CDA, and thus all dilute dogs are unhealthy, or that breeding two dilute dogs together will always result in unhealthy pups. Thankfully, this isn't true. There's no evidence that CDA is caused by dilution inherently. Rather, any dog can have the genes for CDA, but only dilute dogs express CDA. So only dilute dogs actually suffer symptoms from a disease that any dog can have the genes for. Think of it like capital D being epistatic to the genes for CDA. One proposed solution to this is actually to breed dilute dogs together more. If both dilute parents are healthy, it's more likely that their pups will be healthy too, since if they had the genes for CDA, they, as dilutes, would express the disease. If one of the parents was non-dilute, though, there's a risk of them secretly carrying CDA and passing it on to a dilute puppy, making them sick. As of this video, there's currently no knowledge that I could find of CDA's locus and no testing for the gene or genes responsible. Let's hope that more research and testing helps us find a better understanding. Enough lightening alleles can make a red look so damn light that it looks white, and enough darkening alleles can make red look as dark as liver, but it's still just pheomelanin. There's a million bazillion names for each and every shade of red possible, and no one agrees on any of them, but breeders seem to be able to at least agree on calling lighter shades of red less intense and darker shades of red more intense. Now, unlike black-based markings, in which just one gene turns all the way from black to blue, red's intensity is swayed by polygenetics. There's surely a lot, but 5 key loci have been identified as controlling about 70% of red's variation. That's a lot of work those 5 are putting in. The first one is, unfortunately, the only one I could find a letter associated with, the I locus for intensity. We're pretty sure that it's a mutation in the first exon of MFS D12 on chromosome 20, but the article that found it also warns not to rule out post-transcriptional or epigenetic factors as well, and that just because they made some really good progress in understanding the gene doesn't mean that we have it fully figured out yet. Capital I makes a darker, deep intensity, while little I makes a lighter intensity. In particular, little I is associated with diluting dogs to the point where they look basically totally white despite their hair still being pheomelanin-based. It's also incompletely dominant, more on that in a minute. Even more significant to red's intensity than the I locus is a recently discovered gene, I believe found in 2020 because that's the publication date of the article that I found, and it reads to me like they found it. If I'm wrong, I'm sorry. It's a mutation nearby KITLG on chromosome 15. For an idea of how big genes are, nearby here means 152,000 base pairs away from KITLG. That's 152,000 individual ladder rungs. So they're practically neighbors. One of my sources just calls this one KITLG, so I'm also just going to call it KITLG. I put it in the pheomelanin affecting section because it's primarily a pheomelanin gene, but it does also have an effect on eumelanin, just not as intensely. It causes the root of the hair to be lighter than the tip of the hair. This has the effect of making a dog look lighter overall. This is reportedly more noticeable on long-haired dogs because longer hairs have longer roots, and thus the light part is also longer. This can have a very intense effect on red, potentially lightening it up quite a bit. Other polygenes are still very much a factor, do not underestimate polygenes, but it's still pretty damn significant. Check it out, ain't that neat? As for its effects on eumelanin, images provided of KITLG show a sort of grayish, tarnished black rather than something like the D locus dilution does. Again, we're more likely to see lighter colors on longer-haired dogs. It looks like this happens genetically because the locus accidentally duplicated itself. In fact, it did this a bunch of times. The more duplicates are stacked on top of each other in a trench coat to make up the dog's allele, the less light the hair's root will be compared to the rest of the hair, and thus the darker the dog will appear. The less duplicates make up the dog's allele, the more light the root will be compared to the rest of the hair, and the lighter the dog will appear. The study at hand did not examine dominance order. They were just trying to locate the thing on a chromosome. They weren't doing pedigree analysis. And given that it's so new, and also that it's one gene contributing to a pretty big polygenetic gradient trait, I can't find anyone who did examine in dominance order. So I actually have no idea what the dominance relationships are here. The article itself roughly divides dogs into low copy number dogs, who are very lightened by this gene, mid copy number dogs, who are kinda lightened by this gene, and high copy number dogs, who are not lightened by this gene. So for simplified, purely fictional purposes, for now, what I would do personally is make up 3 alleles, a light, medium, and a dark. Since we don't know, and because the I locus's alleles seem to be incompletely dominant, I would say that these are also probably incompletely dominant. But again, all of this is just made up for fictional use with pretend fun family trees. We do not know the dominance order, and no specific alleles have been named that I was able to find. The other 3 of the big 5 red-affecting loci are tire, SLC26A4, and, quote, a link RNA that may be regulating the RUNX gene. I was unable to find more information on these individually. If you have any, please send it to me. I'd love to follow up with more info here. Hey, remember when I said that I isn't completely dominant and that I'd come back to it later? I'm coming back to it now. Apparently little I is epistatic to both KITLG and SLC26A4. This means that if a dog is homozygous little I, we won't see KITLG's diluting effects as little I will overwrite them. Same for SLC26A4's diluting effects, but I don't know enough about the details of how that one works. But a heterozygous capital I little I dog will show both dilution effects, just with I at more intensity than it would be if it was homozygous. I hope to one day be able to give you more information here, but moreover, I hope that talking about these genes has given you a broader understanding of how complicated most genes really are. The majority of genes are like this, and even a lot of the more so-called simple genes are being simplified through years and years of research to make them easier for you to understand, and the reality behind them is often as messy as you saw today. I'm excited to hear more about these loci in the future. One final side note, according to one of my sources for all these red dilutions, Siberian husky breeders call the phenotype that results from a husky having at least one capital I allele Sable. Dog breeders do not communicate. There's a very interesting marking found in Shiba Inus called Urajiro, where the underside of a red dog is lighter, often in a point-like shape. It's suspected that Urajiro has something to do with red-affecting dilution genes, but we don't understand it well enough to say for sure. What we do know is that it only affects red areas, so a dog with both Urajiro and an A-locus marking, like sable, lagodi, saddle, pointed, so on, will only show Urajiro on their red bits, not their black base. The black base covers it, essentially, like a layer above it in Photoshop. The same goes for brindle. A dog can be both brindle and Urajiro, but the Urajiro will only show up on red between the black modeling, again, like a Photoshop layer. I've also noticed that Urajiro seems to only show up on lighter reds, not the super dark ones. I have no idea if that's to do with Urajiro potentially being the result of dilution modifiers, or if it's just breed bias since Shiba Inus, who are known for having Urajiro, tend to be pretty light, or both. At this point, I'm entirely just observing and guessing. Just remember the only appears on red Photoshop layer thing and you're fine. Lastly, there's another form of graying that, as I understand it, isn't technically a dilution at all. It's called progressive graying, as the dog is born black, or liver, and gets grayer, or Isabella-er, as they grow up. Progressive graying is not on the D locus where normal dilution is. That means it's possible for an already blue or already Isabella dog to also have progressive graying, but we'd never see it, just like how we'd never see a black mask on a dog who's already black. That makes normal little d dilute epistatic to progressive graying. Similar to normal D-locus dilute, though, progressive graying only kinda affects red, but unlike dilute, it doesn't affect masks for some reason. We don't know why. So a black dog with progressive graying and a mask starts fully black. You'd never know a mask was there, and then as they age, they turn gray, except for the mask, which stays black. Crazy, right? Progressive graying is represented by G. The only source I have on it says it's proposed in completely dominant, but that it's unconfirmed and hard to prove for reasons that'll make sense soon. Either way, it's capital G, and little g is for a regular dog who doesn't progressively gray. As said, the dog is born black or liver and then lightens as they get older. How light they get and how evenly spread the lightening is across the body both vary a lot, both because the amount of lightening depends on the length of the hair. The thought is, although this is speculation, that the gene is making it harder for hares to keep their eumelanin. So the longer the hair is, the longer it's been growing, and so it's had more time to lose more of its pigment. Longer-haired dogs thus end up lighter than short-haired dogs, and if there's a lot of difference between hair length on different parts of the body, like short on the legs but long in the belly, we'll probably see uneven graying. Given that a lot of long-haired dogs have shorter puppy coats, this might also be at least part of why the graying is progressive with age, but that's a guess from me. If it is incompletely dominant, then that means heterozygous capital G little g dogs don't get as light as homozygous capital G dogs, but I don't think we have a reliable way to guess this from pictures given how individual the effects of the gene are, and I can't find any source with more definitive evidence. I'd say to just go ahead and pick the dominance model that seemed more fun to you until we have more info. Lots of loci covered this time around, but we can only really do Punnett squares for a couple of them since we don't know the letters for Urajiro and the world of red dilution is kind of a hellscape. Those of you who like doing multi-loci Punnett squares, have some fun with dilution and progressive graying. Epistasis is always a blast with multi-loci Punnets. For the rest of you, here's a dilute dog and a non-dilute dog. Excellent. 50% chance of non-dilute and 50% chance of dilute. Now here's two progressively graying dogs. All of their puppies will progressively gray. However, if we're using the incomplete dominance model, there's a 50% chance the puppy will grow into being a little bit lighter than the other 50% chance. We're not done covering eumelanin exclusive modifiers yet. Buckle the fuck up because we're gonna cover Merle next. I'll see you there.